Hello everyone, and welcome to the Synthace DOE Masterclass, DOE for Media Optimization. I'm Markus Gershater, I'm the CSO here at Synthace, and I have the pleasure of being your host for today's session. This webinar is the latest on our series on DOE. All of our previous webinars are available to watch on the website. If you've attended a webinar of ours in the past, welcome back. And if you're joining us for the first time, we're very excited to have you and can't wait to share today's session with you all. So for today's session, I'm really excited to introduce our presenter, Alex Rimmer. Alex is a stem cell scientist at HigherSteaks, where he works on optimizing the growth of stem cells for sustainable meat production. Trained in biochemistry at Nottingham Trent University, he has conducted research into skin development and cancer metastasis at King's College London before joining HigherSteaks in 2021. Thank you for joining us, Alex, and without further ado, I'll hand it over to you.
Perfect. Thanks everyone for coming. Thanks for the little whirlwind back through my career, Marcus. Yeah, Synthace had their own title. I came up with my own, which is Can DOE Replace Piggy? So we are focused at HigherSteaks on something called cultivated meat, which I will run through. So you may have heard it as lab-grown meat, but our idea is we are trying to replace the conventional method of animal husbandry for meat, specifically with pigs. So our product will be bacon and pork belly, hence the awful title. So we're talking about how does DOE improve media development in cultivated meat field. So throughout the talk, I'll kind of cover the basics of like what is cultivated meat, what is DOE, what it's about. I really want to make sure everyone's on the same page. But I promise I will get through to some actual data if that's all you came for. Like that will be there, just hold tight. So why cultivated meat and what is it? So really start off with why cultivated meat exists. And that's because of the global problem of animal husbandry, or the focus of farming animals in order to create meat as a food source. So in 2050, global meat consumption will be double what it was in 2000 due to the increase in population and also the increase in wealth. As people get wealthier, they like to eat more meat. So we're already starting to see the negative impacts of this across the world. And as this increases, it's only going to get worse. So what are some of these impacts? Are the greenhouse gas emissions. So not only the green cows, pigs, everything kind of produce methane. There are other greenhouse gases associated with it. Water overusage. This is particularly prominent in some of the really intense cattle farming that goes on, which is literally draining reservoirs around them. Antibiotic resistance. So about 80% of antibiotics sold were actually for animal usage. And we're already having a developing problem of antibiotic resistant bacteria, and this is only going to make this worse. Lastly, animal welfare. The whole concept of killing animals for meat that we don't really need nutrition-wise is controversial, to say the least. So how can cultivated meat help in all of this? Well, cultivated meat is the process of growing meat without the animal. So what we do is we take an initial sample of cells without killing any animals and then grow this into hundreds of kilograms of meat in bioreactors, taking it out of the animal. So what does this help to solve? Well, water overusage. Our systems are very effective in the amount of water they use. Antibiotic resistance, there is absolutely no antibiotics used in our entire process. Animal welfare, as we'll talk about in a minute, the samples we take don't kill any animals and the animals are harmed as little as is possible. And greenhouse gas emissions. So it's a more efficient process in general, but if we can couple cultivated meat with green sources of energy, such as tidal, wind, solar, we really do end up with a very efficient way of producing meat. So what is the HigherSteaks process? So, as I said before, we take a small sample of primary cells from a pig. We then establish something called a cell line. So this is where we take a sample. We actually produce something called induced pluripotent stem cells. So that is where you take an adult cell and you'll do what you're told throughout, or at least I was told throughout education was impossible, and you turn them backwards into a stem cell. We then are able to grow those stem cells in bioreactors. So the first step, the proliferation stage, that just means to grow as many as possible. So we grow them up to the hundreds of kilograms. Then we differentiate the stem cells or turn them into fat and muscle for our end product, bacon and pork belly. So what's my role? So my focus is medium development. So again, make sure we're on the same page. I'm sure most of you know, but cell culture media is really simply put a sort of replacement for the bloodstream in the cells. So that's a source of energy, amino acids, vitamins, and salts. But there's also a separate part that's especially important in stem cells, and that is these growth factors. So these will increase the amount of cell divisions we can get. But really importantly for stem cells, and really difficult for stem cells, is you want to maintain their stemness or their pluripotency. So we don't want these cells turning into anything else before we're ready. We want them to stay as stem cells. So this is really important for two reasons. One, it's integral to the product, and two, is it's where most of our costs are associated with. So this is a big focus on cost reduction as well as keeping our cell populations high. So why would we look to optimize media at all? There is commercial mediums available. They work. The problem is they're hundreds of times more expensive than we can have in order to scale our product and make it cost effective. They also traditionally don't care if the things in the medium aren't food grade, because no one's going around eating their cells that they're making in the lab. This is a problem for us because that's exactly what we want to do. So we really need to focus on making sure that everything we put in our medium is able to be eaten. And what we have a real big problem is we deal with not only porcine cells, but porcine stem cells. There really isn't a wealth of literature that we can go through to work out what we need in our medium. We don't really have that starting point. What else does DOE specifically help for? Well, it allows us to save a lot of time. So all three of these points I'm going to go into in more detail, but to iterate what it is, it saves a lot of time. It allows us to profile interactions between two factors. So this is really, really common across biology, and it's something you can't do with traditional experimenting. And it also allows us to view this optimization landscape, of which there'll be some nice images later. So here we go. What is DOE? So it is not traditional experimentation. So we know that a lot of people who do DOE know that as OFAT, but that is one factor at a time approach, where you take one condition, you vary that condition, and you change another condition. So, hang on, it's not loading. There we go. So this is the one factor at a time approach on the right-hand side. So let's imagine you just take temperature first off. So we're varying it from 15 to 35 degrees. Some of the guys at Synthace might recognize this one. I did plunder it from one of your resources. Thank you. So you can see across this curve up the top, you checked over 1, 2, 3, 4, 5 conditions, and you found this middle value was the best. Then you ran a completely separate experiment, which changed the pH from something low to something high, and you found, okay, this is the optimum pH and the optimum temperature. That's experiments done. Design of experiments allows you to view this entire landscape across the whole thing and realize, actually, from this gray arrow to this blue arrow, we missed the true optimum from doing one factor at a time approach. So not only did we have to do 2 experiments instead of 1, we didn't get as good as results as we could have done if we did design of experiment. So I'm about to show, it's just a cube with some circles. Nobody stress, I'll explain what it is through, because it looks a bit complicated on the outskirts, but it is really easy. And to try and make it as easy as possible, we're going to use the same things as we did before. So at the bottom, from the left-hand side to the right-hand side, we're varying temperature. So imagine it's 15 to 30 degrees again. Over the other side, we're varying pH again. So this is exactly the same as what we saw before, but we're adding in another factor. So osmolarity, for anyone that doesn't know, is really simply put kind of how many parts there are per volume of water. So That's really simply put what it is. If you can't think what osmolarity is, just think of it as pressure or anything else. It doesn't matter for the sake of this. So each one of these circles on this diagram is a experimental condition. So similar to how we saw on the previous experiment where we varied temperature on the OFAT, we have a condition here which has low temperature, low osmolarity, and low pH on the far left-hand side. Then if we take one step to the right, we have high temperature, low pH, low osmolarity. You get the picture. If I take the far top right-hand corner, we have high pH, high temperature, and high osmolarity. So that's each one of the black circles on the corners. The red circles in the middle are in the middle of the cube, in the center of the cube, and they are center point values. So they're the middle of the road temperature, the middle of the road pH, and the middle of the road osmolarity. So the final thing you can do for a DOE is testing on the outside of your square, and this will basically just let you know, were you in the right range to start off with, or did you set your levels wrong? Really simply put, that is sort of all a DOE is from its entirety. It's a way that we can test 3 things in this example at the same time and test all the different levels. So how does it do this? Well, it's a statistical approach that allows us to screen these 3 factors within the same experiment. The benefits of this: we get to view the full design space. So similar to how we saw on the previous graph, which was only 2 experiments where we missed that optimum value, we can now find that optimum value because we get to test every single point with statistics within this entire cube, essentially. We also, again, we get to test all the 3 factors within the same experiment, which is a big time saving. So lastly, and this tends to get overlooked when you look at initial kind of things about how to do DOE, but it's really one of the most important factors, at least for my work. So I'm going to try and cover it. And this is, so let's strip it back from that cube we saw before. So we only have the points that are low temperature, low osmolarity, and low pH. There's a really fun, interesting thing that design of experiment will let us do, and that's reducing the experimental conditions that we can take. So if all you take from this slide is that we are able to reduce the amount of conditions, so we reduce the amount of experimental runs we do per experiment, that's fine. That's all you need to take away, but I will try and explain it a little bit more. So it's something called a partial factorial, where all we need to do, so let's say we took low temperature, low osmolarity, and low pH as one of our conditions we test. We tested this one up here, the high temperature, high osmolarity, low pH. This one here, I won't run through what the Markus did that before, and this one here. So if we only tested these 4 conditions out of the 8, so we only tested half of the conditions that we could have possibly tested. What have we actually looked at? So have we looked at low temperature? Yes, we looked at low temperature twice. Did we look at high pH? Yeah, we looked at high pH twice. Did we look at low pH? Yes, twice. What about low osmolarity? Again, twice. And high osmolarity? Again, twice. So we can use a statistical approach to actually figure out what the other corners of this square would have been from what we actually tested. So long story short, it basically allows us to test less conditions than we otherwise would have had to. And you'll see in a second why this is really, really, really important for my process. So we're here. Thank you for waiting. But this is now what I actually do at HigherSteaks to develop Medium. So we use a software called Jump. So Jump is basically the software we use to develop the design of experiment. So a design of experiment is quite complicated to do by hand. You can imagine I've got 10 factors over all those conditions we saw. You'll see it later on, but you end up with this big table. That's what Jump does. Synthace, as you all may have heard of, is our way of converting that DOE file into instructions for automation. Then what we do is we actually do the experiment. So this is where we're going to use a liquid handling robot to do the experiment. So what are we looking at? So Jump was this statistical software that I was trying to explain is designing the DOE itself. So what I've done here is I've made up a design of experiment for a 10 different component experiment, so A through K, and it's at levels. So if we imagine this as a concentration, say it's 1 milligram per mil, that's what I'm testing. So it's tested at 0 mg per mil, as in there was none of the concentration. It's tested at 1 milligram per mil, and it's tested at 2 milligrams per mil. So we've got 3 different levels component A has been tested in. You can see each of these levels has been tested numerous times. So that allows us to fill in the gaps, just like I did in the previous screen, in the previous cube. So this is where the power of fractional factorials come in, and why I wanted to explain it. So this experiment here is 25 different conditions for one experiment. If without this fractional factorial, it had been closer to 60,000 experiments. So this is where it's really critical to at least have in mind the power of these fractional factorials. Now, I won't go into all the details, but you do lose something for doing this. There is no free lunch, but it allows us, at least in this screen, it would allow us to say which of these components is doing something useful. It is really powerful for that. So what's the next step? So sorry everyone at Synthace, I'm going to run through this fairly quickly because software does a lot more, but this is what I use it for. So this is what the Synthace software looks like for me. So really simply, at the start, this first box, I put in that horrible looking table. I pop that into here. So I had components from A to K. I would pop those components in here and say what concentrations these stocks were at in this box to find liquids and plates. Then there's a run DOE plate where I would say what my final volume is and what my plate format is, and then it exports it. So why is this really helpful? It's hard to grasp, it's hard to explain, and it might be hard to grasp with my explanation, but there is a huge amount of work to go from that DOE file into plugging it into automation. So what you need to do there is so the DOE file will output a concentration, and the liquid handler will want a number in like a volume number, like microliters. And the jump there means you need to work out your stock concentrations that would work, and then your volume that you need per microgram per mil for each of those steps. So if we imagine our really quick experiment earlier, so that was 25 different conditions and 10 different components. So that's 250 times I need to do those calculations and then input them into the automation software, which is days worth of work potentially, and it's something that Synthace will do very quickly. Lastly, you just run the experiment. I say automation can be really tedious, but it is worth it. It removes the human aspect of pipetting for an entire day. Some of these screens are very big with lots of different components in them. One I'm going to go through next is a very large screen that I just wouldn't have been able to do without automation. The amount of pipetting is bound to lead to mistakes. They're small well plates. There's a lot of pipetting to do in each one of them. And also just no one wants to do that. I wouldn't want to set anyone the task of doing a big screen manual pipetting it. So what does this look like? What's the outcome of all this? We've done all the prelim. What does it actually look like for me at HigherSteaks? So this is a picture of what our first medium was designed to do. And it's designed to create grow stem cells, within a suspension. So these are cells that are floating. So when stem cells are floating and you put them through a slight kind of vortex, a moving liquid, they will have the tendency to clump together. They like to be together. And once they've clumped together, they then grow from there. So what you're looking at here are pictures of clumps of cells that are growing. These clumps of cells with stem cells are called embryoid bodies, but they are just clumps of cells that are growing in suspension. So this is an example of one of the wells in one of my DOEs, and this is an example of another one of the wells within that very same DOE. So when I say wells, these are obviously in small wells within a plate. and each of these wells within that experiment had the different conditions or different media formulations within them as designed from the design of experiment. So what this shows us is the design of experiment worked because there are differences from one condition to the other. If there was no change in the condition, then it wouldn't have worked out very well for us. So what this screen was 22 factors. So 22 different growth compounds within one single experiment. So again, I'm going to harp on about these partial factorials. This screen was around 320 different conditions. If I'd done this screen using without a partial factorial, using a full factorial, it'd have been around 31 million. This is really where there's a huge difference in what you're able to achieve with a partial factorial and design of experiment. Let's test this out versus one factor at a time. So if I did use one factor at a time, each factor, the experimental run would have taken around a week. If I was to test all 22 of those, you're talking 22 weeks, simple maths. So kind of half a year to do one experiment. That's not going to get an okay from the bosses at HigherSteaks. So let's have a look at the analysis. What do you get out from doing a design of an experiment? Well, you get out something like this. So this is where we plugged our results back into the JUMP software, and we get this come out. So obviously I've anonymized all of the conditions here. Sorry for everyone else interested. There is such thing as IP and trade secrets, and I'm heavily bound to them. So all you're going to get is component 1 to 8 for this subset that I've chosen here. So on the axis at the top is essentially our growth rates. So the very low down would be there wasn't many cells growing, and very high up would be the more cells that grew. So let's have a look at component 2. Let's pull it out. So it looks like from this experiment, we may have found a rough optimal. You can see it's at low concentration, cells don't grow as well, and at high concentration, cells don't grow as well, which is kind of to be expected. So it looks like we know roughly where the optimal value lies for that component. How about 8? So 8 to me looks like it may lie slightly to the left-hand side of this, i.e. we have not tested a low enough concentration that is optimal. So, we'd go on to do another experiment where we would change the conditions of that. So I might run it from, let's say, this value down the bottom, we might run it from 150 to 250 to see where it lies within that range. And then, especially with this initial screen, you get a lot of things that look like component 7. So component 7, throughout the whole variation, didn't do anything on the growth of our cells. Adding this into our media had no effect on the growth, and therefore has an increase in cost for no beneficial output. So we remove this from the next screen. So how about the interactions we were talking about? Well, this is a real world example of what these interactions can look like. So this is what you can get with a design of experiment to look at the interactions between two components. So we can see here, it's a little bit less extreme and pronounced than the example we gave at the start. But if we took the maximum value of component 2 and then went ahead to vary the maximum value of component 1, we would miss the optimum value there. So one factor at a time wouldn't have found the optimum concentration for component 1 and component 2. So you can see the interaction profile for all the components. If you try and, I don't know, created a graph representation of all of them together, it wouldn't mean anything. We don't have that many dimensions in graphs. It would start to make nonsense. But you can pull this data out for all the components, the direct interactions between the two components across the whole surface, which is really, really interesting to see. And it really helps with the robustness of your experiment. You're not left wondering, oh, what about the gaps with one factor at a time? So you saw from that initial graph we missed it because we weren't looking at the entire landscape. Whereas here, I can see the entire landscape for all of this, which is really, really powerful for just believing the results of your experiment. It doesn't leave you thinking, oh, what if I'd done this? Like, you have a idea of what it would look like outside of these boxes, because you can kind of see where the curves are going. So what's our entire process of this medium development from start to finish? So I mentioned before that although we had 320 runs out of 31 million, which is incredible for looking at what factors are contributing. So we mentioned one of those components wasn't contributing at all to the growth, so we removed it. It's really, really powerful for that sort of stuff. But because of the lack of data points that we had, the statistical power to resolve where is optimal isn't so good. So it's a trade-off. So after a screen, we would assess which the components that worked good, and we run them through something called a higher power design. So it's again a design of experiment, but it will have essentially more relative points, which allows the statistics to be better, which then allowed us to finalize the media, finding these optimum values. And then we can test it scaled up. So what did this end up looking like? Well, we found we had, in the end, a greatly improved 3D medium at a lower cost, which both increased the cellular yield at the same time as reducing the cost by orders of magnitude from the medium that we were originally using. So this is the stem cell. This is one of the cells from the initial screens I showed you. So as you've seen this very same image. And then the next image is slightly zoomed out, but gives you the concept. So this was our medium after we had finished the optimization process. Under the same conditions, that's how many cells, clumps of cells we got, and at a reduced medium cost. So this is really where design of experiment can kind of come into its own, and you can see the results here. So this whole process, the diagram was slightly simplified, but it took a bare minimum amount of experiments. I think we ran 3 or 4 maybe experiments to get to this final end product. And compared to that time frame for that just initial screen of 22 weeks, that would have missed some of the optimum values and really just wouldn't have been feasible, massively, massively reduced experimental load. And this was possible with a team of 1 and then eventually 2 people. So this is what is achievable with design of experiment. We did not have a big team behind us. We had 1 to 2 people that could make this medium. So we haven't stopped there, and I wanted to run through another one of the mediums we created with design of experiment, because it's a little bit less traditional. It's kind of how theoretically DOE should work versus how this one worked in practice, and what we did to kind of salvage it. So as I'm alluding to, it wasn't a classic design of experiment. So this one was a 2D rather than a 3D medium, and the results from the initial screen were not great. We tested hundreds of different medium conditions, and only around 5 of those different conditions were suitable for cell growth. So we used some of the results from our first screen that I mentioned, the 3D screen. We plugged those in, and we had a different cell line as well. So that was one of the big differences there. But we didn't see the growth that we expected. And it was impossible from this to do a traditional statistics, DOE statistics on this, because we did not have enough conditions to go from. So as we looked at in the initial kind of cube, we needed all of those data points in order to give a good representation of what data points we didn't survey were. And on this, we only got 5 out of 100 of those data points. So there was no way that we could really interrogate our results of that experiment. So this was really a failed design of experiment. But we took learnings from it. So what did we do next? Instead of just scrapping the experiment as a whole and saying, oh, this doesn't work for us, we can't do it, we used the small amount of data we had. We could see what kind of conditions, what concentrations of medium were effective. and we plot that into a second DOE. So this DOE, I managed to use a higher power condition, and during this one, around 90% of the conditions, so instead of 5 out of 100, we had around hundreds, we had around 90% of them were suitable for cell growth, a much better design of experiment. And this experiment actually allowed me to judge optimum values for our cell media conditions. And this is an image from that experiment. So this kind of gray mass is actually made up, it's actually a nuclei stain. So each one of the gray dots is an individual cell. So this cell medium really, really worked for us. So what does this mean? What were the results of all this? Again, we had a commercial media, but this one was capable of maintaining porcine potent stem cells at 100 times lower cost to commercial medium. And there they are. So not only do we have our own commercial medium, which is food grade, we also have one that is hundreds times lower cost to normal commercial medium, all possible because of this design of experiment approach. The whole bunch of these experiments were done over a few iterations, and instead of the kind of months of pipetting and designing, this experiment took us a bare minimum amount of time. Talking in the scale of a couple months rather than couple weeks, couple months rather than time frame of kind of 6 months, maybe a year. So I really want to talk about, I've just described how powerful I think design of experiment is and how it's good. But over my kind of couple years of doing design of experiment, I can understand some reasons why it's not widely used. Mainly of all is I also would have had no idea what design of experiment was unless I was in this job and at this time. Design of experiment was not something that I had any idea about. If someone had asked me, do you do design of experiment? I'd say, yeah, I'm a scientist. I've designed my own experiments for years, which funnily enough is what I hear all the time now. It's not taught, at least at my university, and I've not heard of anyone that ever teaches this within university. It's not something that's typical within academia in general, at least especially in biology. It's something that's growing in industry, in big pharmas, GSK, AstraZeneca, Lonza, but it's still quite a fledgling field for biology. And bearing in mind, it's been around since 1928, something around there. It really is interesting why that is. I think part of the reason I've also seen is it takes quite a while to design these experiments. There's a lot that goes into them. So if I'm testing 22 different factors, I need to work out what I think the values are, the optimal concentrations are. And this means that you will spend maybe a week like designing a single experiment. And when you're working with people that maybe haven't done design of experiment before, that might look like you're not really achieving very much because you haven't run any experiments within a week. And you kind of get that time pressure of, oh, can you just run this quick experiment so that we get an answer, versus sitting down, designing this full scale experiment that will give you way more answers in a quicker time frame, but you have that lag phase of not getting any answers. And I think sometimes that's quite difficult to explain to people who haven't done this approach before. As well as, especially for me, the amount I'm screening is quite heavily based on automation, which isn't accessible to all as well. So that's kind of my story of how I've got on with design of experiment, how I've utilized it, and my overall thoughts about it. And with that, we're through to Q&A.
Alex, thank you very much. That was an absolutely brilliant talk. Really enjoyed it. I love the kind of very pragmatic approach to design of experiments. It's certainly something we try to promote at Synthace as well. It isn't something to be scared of. You had an experiment there where you ran hundreds of conditions and only 5 worked, and yet you can still build on that. And I think sometimes we get a little bit afraid as to whether we're using exactly the right statistical approach, whether we're using experiments right, design of experiments right or not. Obviously, ideally, we would have everything to the highest level of statistical power and rigor. But I think in a pragmatic sense, bearing in mind what's actually possible in a lab day to day, and the kind of complexity of the systems we're looking at, sometimes that kind of pragmatism does come through. It's certainly something I've experienced, and I hope I haven't really annoyed any statisticians on the call by saying that. But yeah, it's certainly my lived experience of doing this kind of thing.
Yeah,
I think
it's kind of a stage where, like, not quite, but just doing the experiments and, like, okay, maybe you missed like the optimum, or like my experiment that only had 5 working out of all of them. It's like you still benefit so much from those experiments. Like, maybe they weren't designed absolutely perfectly, but you get a lot of value from them all the same. So you really can just kind of give it a shot, give it a test.
And I think that's really important. I mean, it speaks into the overall cultural change thing, and you touched on that towards the end as well. And it's funny, as you were saying that, I remember the first time I used DOE in the company I was working at the time, and even though it showed fantastic results, I had to re-justify my use of it every time I wanted to use it, just because people weren't used to, oh, why do you have to go to all that effort? Can't we just do that quick experiment? That really struck a chord with me.
Yeah, it's definitely one that you have to work your way around, for sure.
And for you personally, what were the hardest parts for you, like adopting DOE and sort of altering your mindset shift? What were the kind of mental barriers and maybe the practical barriers that you had to overcome?
Yeah, I think, to be fair, and because I think it's partly because I am so kind of new to this, like, I understand everyone's kind of, oh, can we just do this quick experiment? Because it is really tempting, because the concept of doing that kind of quick experiment that maybe 1 in 100 times finds the actual optimum value and gets you exactly what you want with actually less effort, it is still tempting, and it is hard to, especially when people are pushing really hard for it against you, like, it is hard to just to sit down and say, no, let's do this one experiment properly, versus like, let's just have a random pop at it. I think overall you will absolutely end up with a better and more rigorous product from doing that. So I think that's some of the mental barriers from it. But really you spin that and you say, I can test so many different factors in such a small amount of time. Like, give me, instead of the time frame of give me a week to test something, if you say give me a month to test 22 factors, that is so much more powerful than saying give me a month and I'll take random pot shots 4 times. Like, it really is, you can switch that mindset quite reasonably.
Yeah. But at the same time, though, you've got to take a fair bit of a almost personal scientific risk, don't you? Because, if you do invest all of that time into that big experiment, and then for whatever reason it goes wrong, I think it's worth acknowledging that kind of, risk taking. Risk taking has to be permitted in the place that you're in, right? You have to have a certain amount of permissiveness, stroke, a bit of top cover, to make you feel like, yes, it's okay for us to give this a go.
Absolutely. Yeah, I completely agree. And then practically it is a big step. Like, it's not only am I saying we have to put the time aside, like, I have to put time aside to design every experiment. I was allowed to, and I'm really grateful for the people at HigherSteaks for doing this, but I took a long time to learn what it was in the first place and get my head around it. And you guys at Synthace were super helpful with that, were like integral to that. I wouldn't have been able to do it otherwise. But it really is a kind of professional learning as well, and that does take a lot of time to properly feel like you've got your head around DOE. Like, I'm good at DOE now. I definitely don't know the ins and outs of, like, the academic side of DOE, like, at all. I would say I have a functional working of design of experiment. I wouldn't say I'm truly the expert that could go and write academic papers on it or all that kind of stuff. Like, it's a huge field, and it's quite a complicated field if you're not naturally great at stats, which I'm not. So there's a lot of stuff there, but there is great resources, and you can do it without being a perfect statistician, because I am absolutely not.
Yeah. So we've got a question here from Nicola in the audience, and she's saying, where did you learn DOE? Was your training provided by Jump? Did you take courses? Can you recommend material?
Yeah, absolutely. So I kind of I was lucky enough that some of the guys at Synthace did help me out with that. But there is quite a lot of online resources. I'll plug Synthace again. They've actually done a great job of producing some really, really interesting resources, some on medium development as well, online. As well as, yeah, Jump provide their own resources. There are a couple of kind of non-professional style YouTube videos, but fundamentally to me, there's some really good books actually. DOE Simplified, and then the follow-up book, another type of DOE called RSM Simplified. They're a really good way of getting a grasp of the statistics behind of it. They do a good job of trying to like reduce the jargon for people like me that aren't statistically particularly knowledgeable. And they give you a good, what I describe as like working knowledge of DOE. There are always, the wonderful internet age, there are ways to do it on your own without anyone else's help, I think. But it is a tricky undertaking, and it isn't something that I would say naturally came to me. I can't really get my head around naturally like complex statistics or things like that. So graphs and graphs.
I think you're not alone in that. I mean, especially the non-intuitive nature of multidimensional experimentation. Like just the fact that you can do, cover a space which intuitively would require 31 million experiments with 320 runs. That is completely unintuitive. And again, it speaks to, having to go out on a limb a little bit and just trust the fact that this will work. I think that could be quite tricky. Yeah, I mean, I'll just follow up on the plug. I mean, the whole reason we're putting out these webinars is to help people to go through that process. So Nicola and others on the call, if you want to, do go onto the Synthace website. We've done quite a few of these masterclasses. Essentially, from the very first one where I'm just evangelically talking about why you should use DOE onward, and hopefully they'll give you some ideas about how you can go about stuff. And we've also written a guidebook which you can go through. And then, yeah, I've got a second, what Alex said about DOE Simplified. It's a great book to start with. So I think we've also covered how much theory do we need to understand to do DOE. And then we've got a question here from Willem Jan. He asks, do you have one example where none of your conditions worked, and what do you do then?
I think luckily, I don't have a condition where nothing worked, but I think you've learned, in a lot of ways, you've learned quite a lot. You kind of need to go back in and maybe have a look at the stats side of things, but you have a big design space that you've tested there where nothing worked. So your non-experiment is actually a very big finding in itself, right? That you found out that throughout this massive broad thing, you can imagine yourself doing a bunch of different one factor at a time experiments to find out the one same thing you found out with it. So I'd make sure you go through your experimental record and everything else and make sure everything made sense. Maybe do a little, I'm going to plug it, a little one factor, just to be like, okay, if I do this and replicate it a bunch of times, do I definitely not have any result? Like, is this a true result? It wasn't some part of my experimental procedure that went wrong. This is a genuine result that nothing within this design space works. And then you have to reassess from where you are. Like, you have found a big finding there in itself.
Yeah. And I mean, we have had experience of that at Synthace, and it's not very nice, I have to say, because you put a lot of effort into it. And what we learned from that is essentially you need two things to have some certainty that you can go ahead with a bigger design. First off is you need a good assay. So you need to be able to measure things reliably. And second, you need some kind of a start point. So you have to have some kind of an idea that, yes, we have some conditions that might produce some results, and then you can expand out from there. And if you don't do that, it can lead to quite a lot of heartache because you've just done a massive experiment and none of it worked. I think Alex's point is a very good one, though. And sometimes in science, we don't necessarily have to have an outcome of a certain type. I mean, here Alex does, but we might just be investigating something. And so a definitive negative result can still help us move forward in that project. We just know those things definitively don't work. Now let's move on and try something maybe radically different.
Yeah, completely good. Completely good. And that is actually, you've touched on something that I didn't mention about, which is actually one of the massive things about DOE is that for our starting point, we didn't have one, because all of the kind of there are stem cell mediums that work for us commercially, but they're black boxes. We have no idea what it is. And any kind of published mediums for us didn't work. So we had ideas about where our medium should lie, but we didn't actually have a known medium to start off with. So we couldn't have one factor at a time anyway, because we had no starting position. So like we really were at this from the start of, we do not know what to do here. Like we need to scan, we need to test the area, and that's where some of these scripts came in really useful.
Right. So you were actually using the study as a way of just covering a lot of space as much as possible to try and increase your chances of getting anything at all.
Anything at all. Yeah.
Wow.
And then we can optimize from there.
So we've got one here about oh wow, the questions are coming in thick and fast. I was looking at one and then they all scrolled up. What responses were you looking for in your experiments, and how do you deal with reproducibility? And I think they're referring to noise in the system. Were you using replicates? And that's from Jessica.
Yeah. So if we're getting into kind of DOE design, so like if start off with the easy one, which was what we were looking at. So we were looking for, it's easier in 2D than it is for 3D, but 3D was they're both growth. 3D we had to use a bit of a proxy for growth, but it was essentially cell divisions is what we were looking for our metric. Like how many cells did we start with, how many cells did we finish with? That was our metric. And in terms of replications, you can replicate screens or results if we have a smaller screen that we weren't too sure about and we kind of wanted to replicate it. We have done straight up replications, so like doubled the number of runs we did. And that really gave us confidence in the system, because when we did that, we really didn't improve. If we took each data set on its own and then them together, we really didn't improve our statistics very much for the amount of extra work. So even though when we did do that, we didn't really see a huge amount of increase in kind of statistical resolving power between it, which made us give us quite good confidence that what we were seeing was representative as well. I don't know if Markus, you want to give a bit more detail about how you could design a DOE to encompass that.
Yeah, I mean, like you can incorporate replication, and sometimes it's needed if you have a particularly noisy system. But like, let's just take the example that Alex had of where he was doing a 320 runs and they're looking at 22 different factors. I don't know whether it was, I don't think it was just a 2-level design, that one, but it was 3-level. So if we think you've got about roughly 100 runs at each of those levels for every factor, the way that it's built. So that's the sort of simplest way I can illustrate that. In a lot of ways, you have a lot of inbuilt replication within the experiment for any of the biggest effects that you might be looking at. That's essentially how DOE works. It's stripping a lot away, but at the same time, you have, you know, I mean, what other experiment would you look at hundreds of runs where they're all, you know, at the same level for a particular factor? You know, in a lot of ways, it's very powerful in that regard, and it can give you way better ability to distinguish signal from noise, even when you don't replicate at all. It kind of depends exactly on the system and how many factors you're doing and how many runs.
Yeah, precisely. And to kind of dig into the design bit a bit more, if you did want to have a look at your noise a bit better, you can do something like replicating the center points rather than anything else, which can give you a bit more power there as well, rather than replicating the whole thing.
Yeah, there's all sorts of tricks that you can do in the DOE design, which can help you if you're worried about noise in particular, which is understandable, especially with cell-based systems.
Yeah, for sure.
So now from Sam in the audience, can you tell me a little bit more about Synthace's automation capability? I think this is a critical aspect, having done something similar for MAB's lentivectors, creating the mixtures manually, right? So this is obviously someone who doesn't want to be doing this by hand.
I feel for you. I mean, I haven't had to do it, luckily, at least on this scale, but yeah, this is one of the things I was mentioning that seems outrageously painful. So I feel for you, Sam. So Synthace, especially, I was trying to explain, takes these really big and really complicated, so imagine that 320 run one file of 22 different components at 320 different mixtures, which is an insane amount of pipetting, an insane amount of concentration mapping. And does that for you all in one step. So I was trying to explain kind of what it was again. But you import this file, and my design of experiment file will say component 1 in micrograms per mil, and then it will have 3 different levels. So it might be the 0 micrograms per mil, 1 microgram per mil, and 2 micrograms per mil. I'll then tell Synthace what the stock concentration of component 1 was. It is then able to say, oh, that's your stock concentration. This is the concentration you wanted in your final thing. Okay, you need 2 microliters to do 1 microgram per mil. Then it then does that for every single different component, for every single different concentration you need, and then we'll export that file in the microliters, so the 2 microliters that are needed for 1 microgram per mil. And that file that it exports, you then add on back onto your automation. So the image I showed you in was a Mantis robot that we were using. And you then add that straight into the Mantis. It's read by the Mantis, is in the right format for it. And then you stick your plate underneath and it will run it with the stock concentrations that you gave. Synthace will also point out that if you say you have a stock that's too concentrated and you wanted to add 0.00001 microliters into one well, it would pick out to you and say, hey, your stock concentration is too concentrated. This isn't going to work for the capabilities of that robot. You need to dilute it a bit more. So it takes a lot of the hassle out of the design of experiment operation, let's say.
Thank you. And thanks for the, nice summary of things. It's often quite difficult to put that into simple terms.
I had a reasonable.
Yeah, no, you did. You did exceptionally well. And so then, like the final thing I just wanted to ask about is, how has your use of DOE changed since these early experiments? If you were to go back and do them again, would you do them in the same way, or have you evolved in the way that you're using them? And what would your approach be now?
Yeah, there's definitely been learnings along the way. Some of them are within a lot of the learning. I was in the fortunate place that I was having a hand throughout the process. So like my initial screens weren't terrible. Like they weren't really, really, really bad from the start, but there's definitely tweaks I would have done. So some of the components I added into my initial screen, I believe that they were essentially counteracting themselves. So if there were 2 components along the same pathway, I don't think I had the resolving power within my experiment to judge when one was low and one was high, if they just counteracted each other within it. So it might have said that, oh, this component does nothing because at one level, so it's component 1 and component 2, they both work along the same pathway. And when component 1 was at 0, component 2 may have been at 1. So they kind of counteracted each other.
Right.
And I really think that affected some of my initial screens. So that's something I'm very mindful of when I'm designing experiments now.
So it's basically about how can you resolve almost expected interactions or interactions that maybe in retrospect you could have predicted, and then try and make sure that your design actually takes account for those right in the design phase. Fantastic. Well, Alex, thanks so much for your talk and for all of your answers to your questions. We had a lot of questions come through. If you haven't had your question answered, then apologies, but we will get back to you via email and answer you those directly. I think it's been really good to hear all of these quite complex things and the complex biology put into really simple terms. So thank you very much for that.
Great. And if just before we go, if I give one final plug, I've never done this before, it feels really weird, but I will be hiring for a research assistant and a scientist in the coming summer, likely. So if you are interested, you can find me on LinkedIn or anything like that. Drop me a message about, maybe more about what we're doing here at Higher Stakes, and it'd be great to hear from you.
Yeah, fantastic. I think, very exciting company to work for, and obviously using some of the most cutting edge techniques you can be using. So with that, we'll conclude the Q&A session. After this webinar, we hope you can fill in the survey so we can see how well we've done, and it can help guide us for future content and webinars. If you've missed any parts of today's webinar, don't worry, we'll send you a link to the recording along with other content related to the DOE Masterclass. If you have any more questions about DOE, you can get in touch with us via hello@synthace.com or by filling out the contact us form on our website. Thank you very much to everyone who attended, and thanks once again to Alex for his fantastic talk and Q&A. We hope you enjoyed the talk and look forward to seeing you again at our next webinar.
Thanks so much, everyone.
Cheers.